What if the secret to life itself was written in the stars long before Earth even existed? Picture the universe in its infancy, a swirling canvas of cosmic dust and starlight, where forces beyond our comprehension began stitching together the very first complex molecules. These weren't just random clumps of atoms. They were the blueprints for everything that followed, the building blocks of planets, oceans, and eventually you. It's a story that begins in the coldest, darkest corners of space, where chemistry defied the odds to create something extraordinary. And today, scientists are piecing together the puzzle of how these molecules came to be, uncovering theories so bold they rewrite our understanding of life's origins. Think about the last time you looked up at the night sky. Those distant stars aren't just points of light, they're the crucibles where the universe's first complex molecules were forged. Imagine vast clouds of gas and dust drifting for millions of years until, against all odds, atoms began to bond in ways no one thought possible. Some theories suggest these molecules formed on the surfaces of icy asteroids, while others argue they were crafted in the hearts of dying stars. But here's the twist: these molecules weren't just simple compounds. They were the precursors to DNA, to proteins, to the very essence of life. How did they survive the chaos of the early cosmos? And what does their existence tell us about our place in the universe? That's where things get even more fascinating. Across 30 groundbreaking theories, each more surprising than the last, researchers are uncovering clues that challenge everything we thought we knew. Could lightning strikes in primordial atmospheres have sparked these molecules into existence? Did they hitchhike across the galaxy on comets, seeding life wherever they landed? Or was there something even more mysterious at work, a cosmic recipe we've only begun to decipher? This video is your journey into that mystery, a deep dive into the science, the speculation, and the sheer wonder of how the universe crafted its first complex molecules, and why they might hold the key to life itself. Buckle up, the story of where we came from is far stranger than you've ever imagined. One of the most enduring theories about life's molecular origins takes us back to Earth's earliest days, a time when our planet was a swirling, chaotic nursery of chemical potential. Scientists call it the Primordial Soup Hypothesis, and it paints a vivid picture: vast, warm oceans teeming with simple molecules like methane, ammonia, and water, simmering under a sky crackling with lightning. The idea is that these basic ingredients, zapped by bolts of energy or heated by volcanic vents, began linking together into something far more complex. In the 1950s, two chemists named Stanley Miller and Miller-Urey decided to test this theory in what would become one of the most famous experiments in science. They filled glass flasks with gases they believed mimicked Earth's early atmosphere: methane, ammonia, hydrogen, and water vapor, then sent jolts of electricity through the mixture to simulate lightning. Within days, the flasks turned cloudy with a reddish sludge, and when they analyzed it, amino acids, the building blocks of proteins, had formed spontaneously. But here's where the story gets complicated. While Miller and Urey's experiment was groundbreaking, later research raised questions about whether their setup truly reflected early Earth's conditions. Some scientists argue the planet's atmosphere might have been less ammonia-rich, dominated instead by carbon dioxide and nitrogen, gases far less reactive in generating life's precursors. Others point out that lightning alone may not have been the sole energy source. Ultraviolet radiation from the young sun, or even heat from deep-sea hydrothermal vents, could have played a role. And then there's the puzzle of handedness, the fact that all life on Earth uses left-handed amino acids, while Miller's experiment produced an equal mix of left and right-handed versions. So while the primordial soup idea remains a cornerstone of origins research, it's clear the recipe wasn't as simple as we once thought. What's fascinating is how this theory keeps evolving. Recent studies suggest that even if Earth's atmosphere wasn't exactly as Miller imagined, his core insight still holds. Given the right ingredients and energy, the leap from simple chemistry to life's building blocks isn't just possible, it might be inevitable. Researchers have recreated versions of the experiment with adjusted gas mixtures, even incorporating minerals found near volcanoes or comet impacts, and still watched amino acids form. It hints at something profound. The universe, it seems, has a knack for knitting complexity out of chaos. Whether in Earth's ancient seas or the icy depths of space, the same laws of chemistry could be quietly at work, nudging atoms together toward something greater. While the primordial soup theory imagines life's ingredients brewing under stormy skies, another idea takes us to the crushing depths of the ocean, where scorching water meets freezing darkness. Hydrothermal vents, those towering chimneys spewing mineral-rich fluids from Earth's crust, might seem like unlikely cradles for life's first molecules. But here's the twist: these extreme environments could have offered something early Earth's surface couldn't: stability, energy, and a perfect molecular workshop. Picture labyrinthine rock formations honeycombed with tiny pores, each cavity acting like a microscopic lab where simple compounds gathered, concentrated, and transformed. The heat from the vents wouldn't just cook these molecules, it would circulate them, creating a natural reactor where chemistry had millions of years to experiment. Recent dives to modern vent systems have uncovered something astonishing: complex organic molecules clinging to the mineral surfaces, including amino acids and even lipids, the same molecules that form cell membranes. The vents' porous structures, rich in metals like iron and nickel, likely acted as catalysts, accelerating reactions that would take eons elsewhere. And the temperature gradients around these vents create a Goldilocks zone, not too hot, not too cold, where delicate molecules could assemble without being destroyed. Some researchers even compare these mineral networks to primitive metabolic systems, with the vents' chimneys functioning like prehistoric enzymes, guiding molecules toward greater complexity. But the most compelling part: these conditions aren't unique to Earth. Similar vents likely existed on ocean-covered moons like Europa or Enceladus, meaning this molecular alchemy could be happening right now across our solar system. It's a humbling thought. The same processes that might have sparked life on our planet could still be unfolding in the dark, hidden oceans of distant worlds. So while hydrothermal vents don't answer every question, like how molecules transition from chemistry to biology, they offer a tantalizing clue. Sometimes life's greatest leaps don't happen under open skies, but in the pressurized depths where Earth itself lends a hand. Now, but what if life's ingredients didn't start on Earth at all? Imagine the vast frozen emptiness between the stars, where temperatures hover just above absolute zero and clouds of dust drift like cosmic ghosts. These aren't just specks of dirt, they're microscopic workshops, their surfaces buzzing with chemical activity. Interstellar dust grains, some no larger than a virus, might have been the universe's first chemistry labs. Their icy coatings, laced with simple molecules like water, carbon monoxide, and ammonia, became the perfect stage for a molecular ballet. When ultraviolet light from nearby stars struck these grains, it didn't just illuminate them, it energized them, breaking bonds and forging new ones. Over millennia, these tiny reactions could have built something extraordinary, the precursors to life itself. Scientists have found clues supporting this idea in some of the unlikeliest places. When the Rosetta spacecraft analyzed Comet 67P, it discovered glycine, an amino acid essential for proteins floating in the comet's icy halo. This wasn't just a lucky find, it was a smoking gun. Comets are cosmic time capsules preserving material from the solar system's birth. If glycine could survive in space, frozen onto dust grains for billions of years, it means the building blocks of life might have been raining down on Earth long before our planet had oceans or atmosphere. And glycine isn't alone. Lab experiments simulating space conditions have shown that icy dust grains exposed to UV radiation can produce ribose, a sugar critical for RNA, and even nucleobases, the letters of life's genetic code. The real magic lies in how these grains work. Their surfaces act like molecular assembly lines where atoms and simple molecules stick, meet, and rearrange. The cold of space slows everything down, giving reactions time to unfold with precision. Then, when a passing star's radiation hits, it delivers just enough energy to push those molecules toward complexity without obliterating them. It's a delicate dance, one that couldn't happen in the violent chaos of a young planet's surface. And here's the kicker: these processes aren't rare. Dust grains are everywhere, meaning the universe could be quietly manufacturing life's ingredients in the dark, one icy speck at a time. This raises a staggering possibility. If cosmic dust chemistry is this efficient, the seeds of life might not be unique to Earth. They could be scattered across the galaxy, woven into the fabric of every star system. It's a humbling thought. The same stardust that formed planets and comets might also have carried the blueprints for biology, turning the cosmos into a silent, patient chemist. And as we peer deeper into space, analyzing more comets and distant molecular clouds, we're not just studying the past. We might be glimpsing the universe's quiet, relentless drive toward complexity and our own cosmic origins. Picture this: an ancient shoreline, not of golden sand, but of pitch-black minerals packed with radioactive power. These weren't ordinary beaches. They were cosmic batteries pulsing with energy as their atoms slowly unraveled. Among them, tiny time capsules called zircons, crystals tough enough to survive billions of years, locked secrets inside their structures. And here's the radical idea: the very decay that made these minerals dangerous might have been the spark life needed. Zircons are nature's record keepers. Formed in Earth's fiery youth, they trap uranium and thorium atoms in their crystalline cages, elements that decay at predictable rates, releasing energy over eons. But on those primordial shores, that energy wasn't just fading into the void. It could have charged the surrounding environment, acting like a slow, steady power supply for chemistry's first experiments. When radioactive particles tore through water or organic sludge, they didn't just destroy, they rearranged, ionizing molecules and forging bonds that ordinary heat or lightning couldn't. Lab experiments show something astonishing. When researchers simulate these conditions by bombarding simple chemicals with alpha particles, the same type emitted by decaying uranium, complex organic compounds emerge, including amino acids and carbohydrate-like molecules. The beach setting adds another layer. Tides would have washed over these radioactive sands, creating cycles of wet and dry that could concentrate ingredients. Imagine waves dragging primordial soup across mineral surfaces, each pass giving molecules more chances to interact under that steady hum of radiation. Unlike lightning strikes or vent heat, which are violent and fleeting, this energy would have been relentless but gentle, a constant whisper encouraging atoms to link up rather than a sudden blast that might tear them apart. There's poetic symmetry here. The same process that makes nuclear waste deadly today, the slow breakdown of heavy elements, might have been life's first power source. And zircons tell us something profound. These minerals date back 4.4 billion years, meaning radioactive beaches existed when Earth was still a toddler. We're left with a breathtaking possibility. Life's first steps may not have required dramatic storms or deep-sea fireworks. Instead, the quiet, persistent glow of radioactivity, humming in the grains beneath an alien ocean, could have set the stage for everything that followed. The universe has never been afraid of paradoxes, and what greater paradox than destruction birthing creation? Deep beneath Earth's ancient surface, another unlikely hero emerges in the story of life's first molecules: volcanic glass. Picture the aftermath of a violent eruption, where molten rock cools so rapidly it freezes into obsidian, its surface riddled with microscopic pores and tunnels. These aren't just random imperfections, they're nature's nanoscale test tubes, perfectly sized to cradle and concentrate the ingredients of life. Scientists studying these glassy labyrinths have discovered something remarkable. Their porous surfaces don't just passively house molecules, they actively sculpt them. When researchers recreated early Earth conditions in the lab, mimicking volcanic beaches where hot springs met obsidian-rich sands, something extraordinary happened. Simple molecules like formaldehyde and ammonia, common in Earth's primordial environment, began threading through the glass's intricate channels. The walls of these tunnels, rich in metals like iron and magnesium, acted like molecular workbenches, holding ingredients in place just long enough for bonds to form. Over time, these constrained spaces fostered reactions that would never occur in open water, coaxing amino acids like alanine and glycine into existence. What makes this so fascinating is the glass's dual role. Its pores filter out disruptive elements while its charged surfaces lower the energy needed for chemical unions, essentially giving molecules a gentle push toward complexity. But the real magic lies in the glass's ability to organize chaos. In vast primordial oceans, molecules might drift apart before ever interacting meaningfully. Inside obsidian's nanotunnels, they're forced into close quarters, bumping together repeatedly like commuters in a crowded subway, except here, every collision has the potential to rewrite the rules of chemistry. Some experiments even suggest these glassy structures could have guided the formation of short protein-like chains, acting as templates that dictated molecular arrangements. It's as if volcanoes, often symbols of destruction, secretly provided the architectural blueprints for life's earliest structures. This theory gains even more intrigue when we consider the cosmic perspective. Volcanic glass isn't unique to Earth. It's been found in meteorites and likely exists on volcanic moons like Io. If these jagged, gleaming rocks truly served as catalysts for life's ingredients here, might they have played the same role elsewhere in the solar system? The answer could be locked within the glass itself, each ancient shard a time capsule preserving the universe's stubborn drive to transform simple chemistry into something far more extraordinary. The rhythmic rise and fall of tides might hold one of the universe's most elegant recipes for complexity. Picture ancient shorelines where shallow pools form under the sun's glare, only to be swallowed again by the ocean hours later. This endless dance, water advancing, retreating, evaporating, could have been nature's way of concentrating life's ingredients to critical mass. In these transient tidal pools, simple molecules wouldn't just float aimlessly. They'd be trapped, concentrated, and given relentless opportunities to link up. Laboratory experiments reveal something astonishing. When researchers simulate these wet-dry cycles with basic chemical soups, something remarkable emerges. Strands of RNA, life's molecular cousin to DNA, begin stitching themselves together without any biological machinery to guide them. The magic lies in the physics of evaporation. As water disappears, it leaves molecules behind like stranded passengers on a shrinking island, forced into closer contact. Nucleotides, the building blocks of RNA that might have ignored each other in dilute seawater, suddenly find themselves shoulder to shoulder, their chemical attractions amplified. Then, as new water floods in, it doesn't just redissolve these clusters, it tests their bonds, favoring the strongest connections. Over countless cycles, this natural editing process could have weeded out weak links, gradually favoring longer, more stable molecular chains. It's a stark contrast to theories relying on single, dramatic events like lightning strikes. Here, life's building blocks assemble through patience and repetition, one tide at a time. Recent experiments at the University of Washington took this idea further, mimicking prehistoric tidal pools with mixtures of amino acids and nucleotides. Under cycling conditions, these molecules didn't just form random chains, they began exhibiting a crude version of molecular memory, with certain sequences recurring more frequently. Even more compelling, the wet-dry environment helped solve one of chemistry's trickiest problems: how to remove water molecules during bond formation, a necessary step for creating polymers like RNA. In nature's tidal laboratories, evaporation did this work effortlessly, like an unseen hand guiding atoms toward complexity. But perhaps the most poetic aspect of this theory is its universality. Tidal pools aren't rare miracles. They'd have existed on any rocky world with liquid oceans and a moon to pull them. Mars shows evidence of ancient shorelines, and Saturn's moon Titan boasts methane tides. This means the same gentle, cyclical forces that might have coaxed life into being on Earth could be at work right now on distant worlds, their own molecular symphonies rising and falling with each alien tide. The universe, it seems, has a fondness for rhythms, and in those rhythms, the potential for life may be written in water's infinite ebb and flow. The violence of asteroid impacts might seem like the last place you'd expect delicate molecular building blocks to emerge, but nature has a way of turning chaos into creation. Imagine a space rock the size of a mountain screaming through Earth's early atmosphere at tens of thousands of miles per hour. Its collision wouldn't just leave a scar on the planet's surface. The shockwaves would reverberate through rock and water, generating temperatures hotter than lava and pressures greater than the deepest ocean trenches. In that apocalyptic instant, something remarkable could happen. Simple compounds shattered and reassembled into the complex molecules life would later use as its foundation. Scientists studying ancient impact sites have found traces of this molecular alchemy. Take the Sudbury crater in Canada, where a massive asteroid struck nearly 2 billion years ago. Deep within its fractured rocks, researchers discovered organic compounds clinging to microscopic sulfide minerals compounds that shouldn't have survived such extreme conditions. But here's the twist: the sulfides acted like molecular bodyguards, shielding fragile carbon structures from complete destruction while the impact's energy reshaped them into more robust forms. Laboratory experiments back this up. When teams simulate asteroid strikes by firing projectiles at sulfide-rich mixtures mimicking early Earth's crust, the resulting shockwaves produce amino acids and even components of DNA. It's as if the violence of the impact compressed millions of years of chemistry into seconds, forcing atoms into arrangements that quieter environments couldn't achieve. The real surprise lies in how versatile this process could be. Unlike hydrothermal vents or tidal pools, which require specific settings, asteroid strikes were happening everywhere on the young Earth, and each impact zone became a temporary crucible. Superheated water circulating through fractured rock would have carried dissolved minerals and gases, creating a pressure cooker for organic synthesis. Some models suggest these conditions could have generated not just isolated molecules but entire networks of reactions, like a primordial assembly line operating in the crater's aftermath. Even the soot from wildfires triggered by impacts may have played a role, its carbon-rich particles providing scaffolds for more complex chemistry. This paints a startling picture of Earth's early years. Instead of a serene incubator for life, our planet was a pummeled battlefield, and each catastrophic collision might have been secretly productive. The same forces that could erase life in an instant might also have provided the tools to build it. And if that's true here, imagine the implications for worlds like Mars, where ancient impacts were even more frequent. The universe's recipe for complexity might not just be written in stardust or tidal pools. It could be etched into the very scars of planetary violence, proving once again that creation and destruction are two sides of the same cosmic coin. Among the most unassuming yet powerful candidates for life's molecular midwives are clay minerals, those soft, layered sheets that crumble between your fingers. At first glance, they seem inert, just dusty remnants of weathered rock. But peer closer, and you'll discover these unremarkable flakes possess an almost uncanny talent for organization. Take montmorillonite, a clay so abundant on early Earth it likely blanketed riverbeds and shorelines. under the right conditions, its surface doesn't just passively collect molecules, it actively arranges them like a microscopic foreman directing construction. The secret lies in the clay's structure. Each wafer-thin layer carries a subtle negative charge, attracting positively charged molecules like nucleotides, the very building blocks of RNA. Once trapped, these molecules aren't left to drift. The clay's ridged surface holds them in precise alignment, close enough to bond but not so close they repel. Laboratory experiments reveal something astonishing. When researchers mix montmorillonite with nucleotide solutions, strands of RNA up to 50 units long spontaneously form within hours. Without the clay, those same reactions take weeks or never happen at all. It's as if the mineral provides both a scaffold and a catalyst, its atomic scale grooves serving as templates where chaos gives way to order. But the brilliance of this system goes deeper. Montmorillonite doesn't just accelerate random bonding, it favors specific configurations. Some studies suggest the clay's surface may have selected for RNA sequences capable of limited self-replication, effectively jump-starting a primitive form of molecular evolution. Imagine shallow pools on early Earth, their bottoms lined with these catalytic clays, each tide washing in fresh ingredients while the minerals tirelessly assembled them into ever more complex chains. Over millennia, this quiet, repetitive process could have built molecules sophisticated enough to take the next step, folding into shapes that could copy themselves without the clay's help. What makes this theory especially compelling is its universality. Clays form readily wherever water interacts with volcanic rock, a scenario almost guaranteed on young, geologically active planets. Mars' ancient lakebeds are strewn with clay deposits, and recent probes found them on Ceres and Europa too. This raises a tantalizing possibility. The same mineral that might have guided life's origins here could be doing so elsewhere in the solar system, its unassuming layers patiently orchestrating molecular symphonies in alien mud. The universe, it seems, has a knack for turning the mundane into the miraculous, one humble clay particle at a time. Ultraviolet light might seem like the last force you'd expect to build life's complex molecules. After all, we know it as a destroyer, slicing through DNA and bleaching colors from fabrics. But in the shallow waters of early Earth, this same harsh energy could have been the invisible hand guiding atoms toward complexity. Picture sunlit lagoons no deeper than a puddle, their surfaces shimmering under relentless UV bombardment from a young, unfiltered sun. Beneath that assault, something counterintuitive was happening. Molecules weren't just breaking apart, they were linking together in chains that would one day form proteins. The secret lies in a delicate balance of conditions. While UV light can tear molecules apart, it also delivers precise bursts of energy enough to nudge certain chemical reactions forward without obliterating the products. Laboratory experiments mimicking these primordial pools have revealed an unexpected twist. When amino acids float in shallow water with minerals like titanium dioxide common in volcanic sands, UV light doesn't just randomize them. It drives select amino acids to bond into short peptides, the building blocks of proteins. The mineral surfaces act like molecular dance partners, stabilizing the fragile intermediates long enough for bonds to form. One study at the University of Arizona showed that under these conditions, chains up to 20 amino acids long could assemble in just days, a blink of an eye in geological terms. What's even more fascinating is how the water itself plays a role. Unlike deep oceans where UV penetrates poorly, shallow pools allow light to reach mineral-rich bottoms while keeping temperatures moderate. Evaporation at the surface concentrates the amino acids, increasing their chances of encountering both UV photons and catalytic minerals. It's a Goldilocks scenario, enough energy to drive reactions, but not so much that everything disintegrates. Researchers call this UV-assisted polymerization, and it solves one of biochemistry's biggest puzzles: how early proteins could have formed without the elaborate cellular machinery that builds them today. The implications stretch beyond Earth. Mars once had shallow lakes that would have experienced similar UV exposure, and icy moons like Europa have mineral-rich surfaces bathed in Jupiter's harsh radiation. If UV light could coax peptides into existence here, the same process might have unfolded elsewhere in the solar system. It's a humbling reminder that life's origins may not have required gentle nurturing, but rather a precise type of turbulence, a cosmic forge where destruction and creation danced in perfect sync. Deep beneath Earth's surface, where tectonic plates grind and mountains are born, lies an alien landscape of crushing pressures and searing temperatures, a realm where ordinary chemistry bends to extreme physics. Here, in the planet's stony heart, the sheer weight of overlying rock transforms simple molecules into architectures that would never survive at the surface. Scientists exploring these conditions through diamond anvil experiments where tiny samples are squeezed between the hardest known material have watched methane morph into complex hydrocarbons and nitrogen rearrange into the precursors of amino acids. The pressures in these subterranean labs reach levels found hundreds of kilometers underground, replicating environments where carbon dioxide crystallizes into solid rocks and water becomes a bizarre, electrically conductive fluid. What makes these deep Earth reactions so extraordinary is how they flip conventional chemistry on its head. At surface conditions, building complex molecules usually requires energy inputs like heat or light to overcome repulsion between atoms. But under extreme compression, the rules change. Atoms shoved together by forces equivalent to 50,000 atmospheres begin interacting in ways that defy textbook predictions. In a groundbreaking experiment at the Carnegie Institution, researchers subjected iron, carbon, and water, all common in Earth's mantle, to these conditions. The result, organic molecules resembling fatty acids, the very compounds that form cell membranes. It's as if the planet's interior operated as a gigantic organic factory, its unrelenting pressure acting like an invisible chemist's hand, forcing ingredients together with a persistence no surface environment could match. Even more intriguing are the mineral catalysts lurking in these depths. Certain mantle minerals like olivine and bridgmanite have crystalline structures that align molecules with perfect precision under pressure, almost like molecular assembly lines. Recent simulations suggest these minerals could have guided the formation of peptide bonds, the links between amino acids in proteins, without requiring the water-removing chemistry needed at Earth's surface. Some hypotheses propose that entire networks of organic synthesis might still be occurring today in the deep biosphere, where microbial life thrives thousands of meters below the seafloor. If true, it means Earth didn't just host one cradle of life. It may have nurtured multiple origins in parallel, from sunlit tidal pools to lightless subterranean realms where pressure, not sunlight, provided the spark. This theory reshapes our understanding of life's potential across the cosmos. Ice giants like Neptune and Uranus, with their diamond rain and supersonic oceans, or rocky exoplanets 3 times Earth's mass, could all harbor similar high-pressure chemistry. The universe's recipe for complexity might not require balmy surface conditions at all, just patience, the right mix of elements, and the inexorable squeeze of gravity. After all, if molecules can find a way to organize under thousands of kilometers of rock, what other impossible chemistries might be unfolding in the crushing embrace of distant worlds? On a world where rivers carve valleys not with water but with liquid methane, the chemistry of life might take forms we can barely imagine. Saturn's moon Titan is a frozen mirror of early Earth, complete with rolling dunes, storm clouds, and vast lakes of hydrocarbons. But beneath its orange haze lies a radical theory that lightning crackling through Titan's methane skies could have sparked molecular complexity unlike anything seen on our planet. Laboratory simulations reveal something startling. When scientists zap mixtures of methane and nitrogen, the dominant gases in Titan's atmosphere. With artificial lightning, the resulting gooey substance called tholin contains amino acids, nucleotide bases, and even the pinkish hues observed in Titan's haze. These aren't just random byproducts, they're eerily similar to the organic gunk found in meteorites and comets, suggesting Titan's electric storms might be cosmic chemistry labs in disguise. The process works because Titan's environment turns conventional chemistry on its head. On Earth, water is life's solvent, but on Titan, liquid methane takes that role, and its frigid temperatures, dipping below -180 degrees Celsius, slow reactions to a crawl. Yet when lightning strikes, it superheats methane molecules just long enough to break their stubborn bonds, allowing them to reassemble into more complex architectures. The real magic happens in the lakes themselves. Cassini spacecraft data showed that Titan's liquid methane reservoirs likely contain dissolved nitrogen, creating a perfect soup for electrical discharge experiments on a planetary scale. Imagine bolts of energy ripping through these dark, viscous lakes, each flash forging new molecular connections in the hydrocarbon depths. What makes this theory so compelling is the resemblance between lab-made tholins and the building blocks of terrestrial life. Under microscopes, tholin particles show layered structures resembling primitive cell membranes, and some even exhibit light absorption patterns matching those of complex organic molecules found near hydrothermal vents. Researchers at NASA's Goddard Space Flight Center took this further, simulating Titan's upper atmosphere with UV radiation instead of lightning, and still produced adenine, one of DNA's key nucleobases. It hints that whether through lightning or sunlight, Titan's chemistry is predisposed to complexity, even in conditions we'd consider utterly hostile to life. The implications stretch far beyond one icy moon. Exoplanets with thick methane atmospheres could host similar processes, their own electric storms quietly assembling prebiotic molecules in alien skies. If Titan's methane lakes can manufacture these compounds today, 4 billion years ago, when the solar system crackled with more energy, similar environments might have seeded countless worlds with life's raw ingredients. Titan's greatest lesson might be this: the universe doesn't need Earth-like conditions to build complexity. Sometimes all it takes is a spark in the darkness and patience as vast as the cosmos itself. Far from the warmth of any star, in the frozen voids between solar systems, ice clings to dust grains like microscopic winter coats. These aren't just ordinary frost particles. They're cosmic safe rooms where fragile molecules huddle together, protected from the harsh radiation that would tear them apart in open space. Scientists studying these icy grains have uncovered something astonishing. Their frozen layers don't just preserve simple compounds, they actively rearrange them into the building blocks of life. The European Space Agency's Rosetta mission found the smoking gun. When analyzing Comet 67P, they detected glycolaldehyde, a simple sugar essential for forming RNA, trapped in the icy matrix. This discovery shook the scientific community because it proved that the universe's coldest corners aren't chemical deserts, they're organic factories. The magic lies in the ice's structure. At temperatures barely above absolute zero, water molecules freeze into open, cage-like formations, creating microscopic pockets where other compounds like methane, ammonia, and carbon monoxide get trapped. When ultraviolet light from distant stars hits these grains, it doesn't instantly destroy the ice. Instead, the energy gets absorbed and redistributed, breaking some bonds while gently nudging others to form. Laboratory experiments simulating these conditions show something remarkable. Over time, these irradiated ices produce complex molecules like amino acids and even the nucleobases found in DNA. The ice acts like a molecular sculptor, its crystalline framework holding ingredients in precise orientations that favor specific reactions. One NASA study zapped icy mixtures with proton beams to mimic cosmic rays, and within weeks the ice darkened with organic compounds resembling the tar-like substances found in meteorites. But the real breakthrough came when astronomers pointed radio telescopes at young star systems still swaddled in their birth clouds. In the constellation Ophiuchus, 400 light-years away, they detected the chemical fingerprints of ribose and other sugars swirling around infant suns. These molecules weren't forming on planets, they were assembling in the frigid outskirts of protoplanetary disks, where temperatures hover around -260 degrees Celsius. The implication is staggering. The sugar molecules needed to build RNA and, by extension, life, might be a standard byproduct of star formation, woven into the fabric of every young solar system. What makes ice grain chemistry so compelling is its universality. These frozen specks are everywhere, in comet tails, interstellar clouds, even the rings of Saturn. Their surfaces provide both a shield and a stage, where cosmic rays and starlight play the role of matchmaker, introducing molecules that would never interact in warmer, busier environments. It's a quiet, patient process, one that could have been brewing life's ingredients for billions of years before Earth even existed. And if that's true, then the sweetest irony of all is this: the universe's first complex molecules might not have been forged in fire, but in ice, a delicate dance of chemistry at the edge of frozen darkness. Imagine early Earth's oceans not as the slightly acidic waters we know today, but as vast, alkaline baths, chemical powerhouses where molecules assembled with astonishing efficiency. This theory hinges on a simple yet profound shift in perspective. What if life's building blocks didn't just tolerate high pH environments, but actually required them to form? Alkaline vents, those towering mineral chimneys spewing warm, mineral-rich fluids into ancient seas, might have been the universe's first molecular workshops. Unlike today's black smokers, these vents would have been gentler, their chemistry driven not by extreme heat but by the subtle dance of protons and electrons in alkaline water. The magic lies in the iron-sulfur minerals lining these vents. Picture labyrinthine structures called chemobricoleurs, a French for chemical tinkerers, their porous walls riddled with microscopic compartments rich in iron and nickel sulfides. These metals aren't just bystanders, they're dynamic catalysts capable of shuffling electrons like molecular currency. In laboratory experiments simulating alkaline vent conditions, researchers have watched these minerals facilitate reactions that turn simple molecules like carbon dioxide and hydrogen into acetate, a key intermediate in metabolism. Even more striking, the same mineral surfaces can stitch together amino acids into short peptides under alkaline conditions, a feat far harder to achieve in neutral or acidic waters. One of the most compelling pieces of evidence comes from modern analogues like the Lost City hydrothermal field in the Atlantic. Here, towering white vents built of calcium carbonate, not black sulfides, spew alkaline fluids at temperatures hospitable to delicate organic molecules. The field's pH hovers around 9 to 11, similar to household ammonia, yet it teems with microbial life. Crucially, the chemistry here mirrors processes thought to occur in Earth's infancy. Hydrogen and carbon dioxide react to form organic compounds, powered by the natural proton gradient between the alkaline vent fluids and the more acidic ocean. This gradient isn't just a curiosity, it's a primitive version of the energy harnessing system modern cells use to generate ATP, life's universal energy currency. But the real elegance of this theory lies in its universality. Alkaline vents don't require rare ingredients or special conditions, just water, rock, and the geothermal heat present on any young, geologically active world. Mars's ancient crust shows signs of similar mineral formations, and Jupiter's moon Europa might harbor alkaline vents beneath its icy shell. Even more intriguing, iron-sulfur clusters remain central to life's chemistry today, nestled in the heart of enzymes that power every living cell. It's as if life, once stumbling upon this efficient chemical pathway, never let go, preserving a 4-billion-year-old testament to its alkaline origins in the very machinery of our bodies. The implications are profound. If alkaline seawater truly provided the cradle for life's first molecules, then the search for life beyond Earth might focus not on familiar, neutral pH oceans, but on the stark, mineral-rich waters where chemistry leans toward the basic. In the silent, sunless depths of alien worlds, the same gentle gradients that shaped life here could be quietly at work, proving once again that the universe's recipes for complexity are as diverse as they are ingenious. Now picture this: sprawling geothermal fields stretching across early Earth's surface like a vast, bubbling circuit board, each hot spring, fumarole, and mud pool acting as a node in nature's first organic network. While isolated hydrothermal vents get much of the attention, these interconnected geothermal playgrounds could have offered something even more powerful: a system. Think of it like Earth's original internet, where molecules didn't just form in one place, they traveled, mixed, and evolved across an entire landscape of chemical opportunity. Modern analogs like Yellowstone's geothermal basins show us how this might have worked. In places like the fountain paint pots, acidic hot springs bleed into alkaline pools just meters away, creating a mosaic of pH gradients. Silica-rich waters from one vent mingle with iron-heavy flows from another, sparking reactions that wouldn't occur in isolation. Researchers analyzing these zones have found something remarkable. The diversity of organic compounds here dwarfs what any single vent could produce. Amino acids form in the alkaline zones, then get swept into acidic areas where they link into peptides. Sulfur compounds forged in superheated depths migrate to cooler pools where they stabilize delicate structures. It's a molecular assembly line spread across acres, with each microenvironment adding its own specialized tweak to the recipe. The real genius lies in how these networks could have solved one of abiogenesis' biggest hurdles: concentration. In vast oceans, molecules dilute to uselessness, but in geothermal fields, evaporating pools act like natural concentrators. Steam carries volatiles to new locations, while heavier organics accumulate in shallow basins. Computer simulations at the University of Glasgow revealed that even simple flow patterns between interconnected pools could have created molecular traffic jams, trapping and concentrating key ingredients. Over time, this constant shuffling and concentrating might have allowed rare combinations to emerge like a lottery where the odds improve with every ticket drawn. But the most tantalizing clue comes from the fields themselves. At New Zealand's Waitapu geothermal area, scientists discovered mineral formations called silica gardens, towering, hollow structures that grow when hot alkaline solutions meet cooler acidic waters. These porous towers aren't just geological curiosities. Their intricate channels resemble the microstructure of modern cell membranes. Experiments show they can trap and organize organic molecules, even facilitating simple metabolic reactions. Imagine an early Earth dotted with thousands of these natural laboratories, their steaming networks exchanging not just heat and minerals, but the very precursors of life. This shifts our perspective dramatically. Instead of hunting for a single cradle of life, we might be looking at an entire nursery, a planetary-scale incubator where molecules had countless chances to experiment, fail, and occasionally stumble upon something revolutionary. And if Earth's geothermal networks could host such complexity, why not those on Mars' ancient Tharsis bulge or the volcanic plains of Venus before its greenhouse runaway? The universe may not have bet on one pot to cook life's first molecules. It might have built a whole kitchen. High above early Earth's surface, a thick, orange haze might have been quietly brewing life's ingredients, not in the oceans or vents below, but in the turbulent skies themselves. Picture a planet shrouded in photochemical smog, a global blanket of organic molecules churned up by relentless ultraviolet radiation. This wasn't pollution as we know it today. It was a cosmic kitchen where simple gases like methane and nitrogen collided, split apart, and recombined into something far more intricate. The proof lies not just in computer models, but in our solar system's own time capsule, Titan, Saturn's moon, wrapped in its iconic orange gloom, shows us how such atmospheric chemistry operates on a planetary scale. Its skies are filled with Tholen's complex organic polymers that form when sunlight breaks apart methane and nitrogen, creating a raining drizzle of prebiotic goo. Lab experiments here on Earth have recreated Titan-like conditions with eerie results. When scientists simulate early Earth's atmosphere, packed with methane, carbon dioxide, and nitrogen, and blast it with UV light or electrical discharges, the same tar-like tholins emerge. But here's the twist: analyze that sludge closely, and you'll find it's laced with amino acids, nucleotide bases, and even sugars. It's as if the atmosphere was a giant reactor, its upper layers acting like a celestial distillery where energy cracked simple molecules apart, only for them to reassemble into life's precursors as they drifted downward. The smog layer wouldn't just create these compounds, it would concentrate them with heavier particles clumping together until they grew large enough to fall like organic snowflakes into the oceans below. This process solves two major puzzles at once. First, how did fragile organic molecules survive Earth's violent early environment? High in the smog layer, they'd be shielded from volcanic heat and meteor impacts by kilometers of buffering atmosphere. Second, how did they accumulate in meaningful quantities? Unlike tidal pools or vents which are localized, a global haze would rain its bounty across the entire planet, seeding every lake and ocean with a steady supply of raw materials. NASA experiments with tholin-rich mixtures reveal something even more provocative. When these organics land in water, they self-assemble into hollow, cell-like structures. Some even exhibit primitive membrane properties, selectively allowing molecules in and out, a ghostly echo of modern cells' sophisticated walls. The implications stretch beyond Earth. Exoplanets with hazy atmospheres, like those recently spotted around red dwarfs, could be running similar chemistry right now. And Titan's thick organic rains prove this isn't just theoretical, it's a process the universe clearly favors. Perhaps the most humbling thought is this: the same photochemical reactions that today create urban smog might once have been the driving force behind life's origins. The universe, it seems, has a way of turning its darkest clouds into the brightest beginnings. Cosmic rays, those high-energy particles racing through space at nearly light speed, might have been the universe's original molecular matchmakers. While we often picture Earth's early chemistry as a ground-level affair, the real action could have been happening miles up, where these charged particles from distant supernovas slammed into the atmosphere. Here's how it works. When a cosmic ray collides with a nitrogen molecule in the upper air, it doesn't just bounce off, it blasts the nitrogen apart, leaving behind highly reactive fragments. Studies at the University of Tokyo simulating these conditions found something remarkable. In ice samples bombarded with proton beams mimicking cosmic rays, nitrogen atoms broken free from their molecular bonds readily teamed up with hydrogen to form ammonia, a key ingredient for life's molecular machinery. But cosmic rays don't stop there. Beneath Earth's frozen surface, these particles could have turned entire ice sheets into organic factories. Picture a glacier under constant bombardment, its crystalline structure acting like a trap for broken molecular pieces. As cosmic rays tore through the ice, they not only shattered simple compounds, but also provided the energy to stitch the fragments back together in new, more complex arrangements. Researchers at the Berkeley lab recreated this process by irradiating mixtures of water, ammonia, and methanol common ices in space, with gamma rays the result. Glycine, the simplest amino acid, emerged in the irradiated samples, proving that cosmic rays could have forged life's building blocks even in the most frigid, seemingly inhospitable environments. The implications are staggering. Unlike lightning or UV light, which are tied to specific planetary conditions, cosmic rays permeate the entire galaxy, meaning this chemistry could occur anywhere with an atmosphere or surface ice, Mars' polar caps, Europa's frozen crust, even exoplanets orbiting quiet red dwarf stars. Data from the Rosetta mission supports this. Comet 67P's ice contained patches of organic material precisely where cosmic ray exposure was highest. It's as if the universe, unwilling to leave life's origins to chance, seeded countless worlds with an invisible rain of energy, turning ice and air into cosmic petri dishes where molecules had no choice but to grow more complex. Perhaps most intriguing is the timescale. While other energy sources like lightning strikes or volcanic heat are fleeting, cosmic rays have bombarded every corner of the solar system for billions of years without pause. They're the ultimate patient chemists, working slowly but relentlessly, their experiments running continuously in the background of cosmic history. And if that's true, then the same invisible forces that light up auroras today might have once lit the spark for life itself, not with a dramatic flare, but with the quiet persistence of particles arriving from the void. Phosphorus rarely gets the spotlight in origin of life discussions, yet this unassuming element may have played one of the most critical roles in assembling life's first complex molecules. While carbon, hydrogen, and oxygen dominate modern biochemistry, phosphorus brings something irreplaceable to the table: the ability to link molecules into long, stable chains. Picture early Earth's landscape, where most phosphorus was locked away in insoluble rocks, useless for delicate prebiotic chemistry. Enter schrebersite, a metallic mineral found in iron meteorites that changes everything. When these space rocks slammed into our young planet, they didn't just deliver raw materials, they brought phosphorus in a uniquely reactive form, one that could dissolve in water and participate in the molecular dance of life. Laboratory experiments reveal why this mattered so profoundly. Unlike terrestrial phosphate minerals that stubbornly resist chemical interaction, schrebersite corrodes easily in water, releasing phosphorus ions ready to bond with organic compounds. Researchers at the University of Arizona mimicked ancient meteorite impacts by grinding schrebersite into watery solutions containing simple organics. Within days, the mixture produced phosphorylated molecules, compounds where phosphorus atoms bridge organic structures, much like the backbone of DNA. Even more striking, these reactions occurred without the extreme heat or pressure typically needed to force phosphorus into biological roles. It's as if the universe packaged this crucial element in a perfect delivery system, ensuring it would be bioavailable the moment it touched water. But the story doesn't end with meteorites. Recent explorations of alkaline vent systems uncovered another phosphorus surprise: naturally occurring phosphide minerals in their chimney structures. These deep-sea formations, rich in iron and nickel, can catalyze phosphorus reactions under conditions mimicking early Earth's oceans. When scientists simulated vent environments with phosphides present, they observed the spontaneous formation of pyrophosphate, a molecule remarkably similar to the energy-carrying ATP used by all living cells today. This hints at an elegant convergence. Meteorites may have seeded Earth's surface with reactive phosphorus, while hydrothermal systems provided the perfect workshops to incorporate it into life's emerging chemistry. What makes phosphorus scenarios so compelling is their specificity. Modern cells use phosphorus not just for DNA, but for energy transfer, cell membranes, and metabolic regulation. Its versatility is unmatched. The fact that rare minerals like schrebersite could bypass Earth's phosphorus scarcity suggests a cosmic intervention, as if the universe recognized this element's unique value and ensured its delivery. And if that's true, then planets without frequent meteorite showers or active geology might face a phosphorus famine, potentially explaining why life's spark seems so rare. The search for schrebersite on Mars and icy moons now takes on new urgency, because where this unassuming mineral lands, the path to complexity might follow. Beneath the ocean's surface, where plumes of sulfur-rich fluids billow from hydrothermal vents, lies a clue to one of biochemistry's deepest mysteries. Sulfur, often associated with the smell of rotten eggs, might have been the unsung hero in the story of life's origins. The sulfur world hypothesis flips conventional wisdom on its head. Instead of carbon or phosphorus taking center stage, this theory suggests that humble sulfur atoms, with their unique electron-hungry properties, provided the scaffolding for life's first molecular dances. Modern cells still carry echoes of this ancient chemistry. Deep inside enzymes like ferredoxin, iron-sulfur clusters act as molecular switches, shuttling electrons with breathtaking efficiency, a relic perhaps of biochemistry's earliest days. These iron-sulfur clusters aren't just biological oddities, they're time capsules. In lab experiments simulating ancient vent environments, researchers have watched as iron and sulfide ions spontaneously assemble into clusters identical to those found in living cells today. The process is startlingly simple: mix iron-rich water with hydrogen sulfide under moderate heat, and these nanoscale structures self-assemble like molecular Lego blocks snapping together. What makes them so special is their versatility. A single cluster can store or transfer electrons, stabilize free radicals, even help stitch together carbon chains, functions that would have been invaluable for proto-metabolism. Some scientists liken them to prehistoric batteries, storing geothermal energy in chemical bonds long before cells evolved to do the same. The real power of the sulfur world lies in its ability to bridge geology and biology. At hydrothermal vents, where superheated, mineral-laden fluids meet cold seawater, iron sulfide precipitates form porous, honeycombed structures. These mineral matrices, pockmarked with microscopic chambers, could have served as nature's first test tubes, their surfaces lined with catalytic iron-sulfur nodules. Experiments at the University of Düsseldorf show these mineral compartments can concentrate organic molecules while protecting them from degradation, creating ideal conditions for complexity to emerge. Even more compelling, the pH gradient between acidic vent fluids and alkaline seawater mimics the proton gradients modern cells use to generate energy. It's as if the vents were performing a crude version of cellular respiration billions of years before cells existed. What's often overlooked is sulfur's role as a molecular matchmaker. Unlike other elements, sulfur can form bonds that are both strong enough to stabilize structures and weak enough to allow rearrangement, a Goldilocks scenario for evolving chemistry. Studies of volcanic gases reveal sulfur dioxide's knack for facilitating reactions between nitrogen and carbon compounds, potentially knitting together key precursors for amino acids. On early Earth, with volcanic activity orders of magnitude greater than today, these reactions could have produced staggering quantities of prebiotic material. The Murchison meteorite, packed with sulfur-bearing organic compounds, shows this chemistry isn't limited to Earth. It might be a universal pathway written into the fabric of rocky worlds. Perhaps the most tantalizing evidence comes from extremophiles microbes thriving in today's sulfur-rich hellscapes like hot springs and deep-sea vents. These organisms don't just tolerate sulfur, they depend on it, using iron-sulfur proteins for everything from energy production to DNA repair. Their biochemistry might be a living fossil, preserving metabolic strategies dating back to life's very beginning. If that's true, then sulfur wasn't merely a participant in life's origins, it was the stage, the director, and the script all rolled into one. And as we search for life beyond Earth, these yellow-stained, electron-shuffling molecules might be the signposts we've been overlooking, quietly pointing the way to understanding how chemistry became biology. Among the countless molecules that could have formed on early Earth, one stands out as uniquely capable of bridging chemistry and biology: RNA. This versatile molecule, able to store genetic information like DNA and catalyze reactions like proteins, sits at the heart of what scientists call the RNA world hypothesis. But here's the puzzle. How did ribonucleotides, RNA's building blocks, assemble themselves without the sophisticated cellular machinery that produces them today? Recent breakthroughs suggest the answer might lie in chemistry so elegant it could rewrite our understanding of life's origins. Picture a shallow pool on early Earth, its edges crusted with evaporite minerals that glisten under the sun. In this mundane setting, something extraordinary was possible. Scientists at the MRC Laboratory of Molecular Biology demonstrated that when simple molecules like cyanamide and phosphate are concentrated by evaporation, they can drive the formation of ribonucleotides through a series of self-reinforcing reactions. The minerals don't just passively watch, they actively participate, their surfaces aligning molecules in precise orientations that favor bonding. What makes this so remarkable is how it sidesteps a long-standing obstacle. For decades, researchers struggled to explain how ribose RNA's sugar backbone could stabilize long enough to link with nucleobases. The evaporation scenario solves this neatly, creating fleeting pockets of dryness where these fragile unions become possible. But the real game-changer came from studies of wet-dry cycling, the same process that might have helped assemble RNA strands. Researchers at the Scripps Research Institute discovered that when nucleotide precursors are subjected to repeated drying and rehydration, something unexpected happens. Not only do the nucleotides form, but they begin linking into short RNA-like chains without any enzymes to guide them. Even more astonishing, these primitive strands exhibit a crude form of self-replication, pairing with complementary sequences in a process resembling modern RNA's behavior. It's as if the environment itself was acting as a catalyst, imprinting the logic of replication onto lifeless chemistry. Some of the most compelling support comes from space. Analyses of meteorites like Murchison reveal they carry not just amino acids, but nucleobases like uracil and cytosine RNA's informational components. When combined with ribose-forming reactions observed in interstellar ice analogues, a tantalizing picture emerges. The universe seems predisposed to manufacturing RNA's ingredients, then delivering them to planets via comets and meteorites. Laboratory experiments at NASA Ames Research Center took this further, showing that ultraviolet light shining on these space-borne compounds can push them toward even greater complexity. The implication is staggering. RNA's precursors might have been raining down on Earth pre-assembled, giving our planet a head start in the molecular arms race. What ties these discoveries together is their shared theme of molecular inevitability. Whether through evaporating ponds, meteorite deliveries, or the gentle pulse of tidal cycles, the path to RNA appears less like a series of lucky accidents and more like a chemical destiny written into the fabric of matter. This doesn't just bolster the RNA world hypothesis, it suggests the universe might be hardwired for complexity, with RNA as one of its preferred blueprints. And if that's true, then the same molecular dance that began here could be unfolding right now on countless worlds, their own tides and minerals quietly assembling the threads of life. Imagine a turbulent early Earth where fragile molecular building blocks faced constant threats, crushing waves, scorching heat, and deadly radiation. Survival for these delicate compounds depended on finding shelter, and nature may have provided the perfect refuge, microscopic bubbles made not of soap but of lipids. These fatty molecules, which today form the protective walls of every living cell, could have served as life's first fortresses. Experiments recreating prebiotic conditions reveal something astonishing. When simple lipids like fatty acids are mixed with water and agitated, think waves crashing on a primordial shore, they spontaneously curl into hollow spheres, their water-loving heads facing outward and their water-repelling tails huddling inside. These aren't just inert droplets, they're dynamic enclosures that actively sort and concentrate molecules, creating pockets of order in a chaotic world. One of the most striking demonstrations comes from work at Harvard University, where researchers combined oleic acid, a simple lipid likely present on early Earth, with magnesium ions and heat. Without any guidance, the mixture formed stable vesicles that could grow, divide, and even maintain internal chemistry distinct from their surroundings. Some vesicles developed asymmetrical membranes resembling the polarized structures seen in modern cells. Others entrapped RNA strands, protecting them from degradation while allowing nutrient exchange. It's as if the lipids knew their role, self-assembling into structures that blurred the line between chemistry and biology. What makes lipid bubbles so compelling isn't just their protective abilities, it's their selectivity. Unlike random clumps or mineral surfaces, lipid membranes regulate molecular traffic. Experiments with protocells show they can concentrate amino acids and nucleotides while excluding harmful compounds, effectively curating their internal environment. The University of California, Santa Cruz took this further, demonstrating that temperature fluctuations common in tidal pools or near hydrothermal vents could drive primitive vesicles to actively harvest new lipids from their surroundings, growing until mechanical stress caused them to divide. This rudimentary replication hints at how lifeless chemistry might have taken its first steps toward Darwinian evolution. The implications stretch beyond Earth. The Cassini mission detected lipid-like molecules in the plumes of Enceladus, suggesting similar self-assembly could be occurring in that moon's subsurface ocean. Meanwhile, lab simulations of interstellar ice chemistry show that radiation can transform simple hydrocarbons into amphiphiles, molecules with the same water-loving and water-hating properties that make lipids so versatile. It's a humbling realization. The same processes that built life's first containers on Earth might be unfolding right now in alien oceans, their oily membranes shimmering in the darkness, waiting to cradle the next experiment in cosmic chemistry. Perhaps most remarkable is how these bubbles bridge the gap between isolation and interaction. A lipid vesicle isn't a prison, it's a semi-permeable stage where molecules can interact without drowning in the vastness of the open ocean. In modern cells, this compartmentalization allows for specialized chemistry. In prebiotic times, it may have allowed rare reactions to persist long enough to matter. The universe, it seems, has a gift for creating safe spaces, tiny, oily sanctuaries where randomness could yield to order, and chemistry could whisper its first secrets to the void. Beneath the churning tides of early Earth's oceans, an invisible force might have been quietly guiding molecules toward complexity. Magnetite, that unassuming iron-rich mineral we often find clinging to fridge magnets today, could have been nature's original molecular architect. Picture tiny flecks of this magnetic crystal scattered across volcanic sands, each grain acting like a microscopic compass needle under the influence of Earth's ancient magnetic field, a field 3 times stronger than it is now. When researchers at the University of Tokyo recreated these conditions in the lab, something remarkable happened. Simple organic molecules dissolved in water began aligning themselves along the magnetic fields generated by magnetite particles, their orientations shifting like iron filings near a magnet. But this wasn't just a passive alignment, it dramatically increased the chances of those molecules linking into longer chains, the kind needed to build polymers like proteins or RNA. The brilliance of this system lies in its simplicity. Magnetic fields don't require heat, light, or chemical energy, just the natural pull of Earth's geomagnetic forces. In experiments where scientists simulated tidal zones with magnetite-rich sediments, amino acids subjected to these fields formed peptide bonds up to 10 times faster than in control samples. The magnetite didn't just organize the molecules, it stabilized the transitional states during bond formation, effectively lowering the energy barrier for polymerization. It's as if the mineral provided both the scaffolding and the tools, turning random chemistry into something resembling a controlled assembly line. What makes this theory especially compelling is how it intersects with other origin of life scenarios. Hydrothermal vent fluids are often rich in iron sulfides, which can transform into magnetite under certain conditions. Imagine vent outflow channels lined with these magnetic minerals, their fields directing the flow of organic molecules like traffic controllers at a molecular intersection. Even more intriguing, some meteorites carry magnetite crystals arranged in intricate chain structures so orderly they were once mistaken for fossilized bacteria. If space rocks delivered magnetite to early Earth, they might have seeded the planet with ready-made molecular organizers. The implications stretch beyond our own planet. Mars once had a robust magnetic field and still retains magnetite in its soil, while Jupiter's moon Ganymede generates its own magnetic field through a liquid iron core. If alignment by magnetic minerals truly gave life's molecules a head start, then worlds with active geology and remnant magnetism could be hiding the same quiet catalyst that helped chemistry cross the threshold to biology here. The universe, it seems, never misses an opportunity to impose order, even when the tools are as subtle as the whisper-thin tug of a magnetic field on a molecule barely visible to the naked eye. When we think about life's origins, deserts rarely come to mind, yet these parched landscapes might have been the perfect crucibles for forging life's first complex molecules. Picture an ancient world where vast stretches of land baked under relentless sunlight, their surfaces cracked and thirsty. But deserts aren't just barren wastelands, they're dynamic environments where extreme dryness gives way to fleeting wetness, a rhythmic dance that could have turned these regions into molecular workshops. Unlike the steady conditions of oceans or tidal pools, deserts offer something unique: intense dry-wet cycling, where rare downpours drench the ground, only for scorching heat to evaporate the water hours later. This relentless alternation isn't just harsh, it's transformative, concentrating and reorganizing molecules with each cycle in ways that gentle, stable environments never could. The key player in this desert drama isn't water or heat alone, it's silica, the same mineral that forms sand. Under a microscope, silica grains reveal a secret. Their surfaces are pocked with microscopic pores and ridges, perfect for cradling fragile organic compounds. When water briefly floods the desert, it carries dissolved molecules like amino acids into these tiny cavities. Then, as the water vanishes under the sun's glare, those molecules aren't left to degrade. They're sheltered by the silica's structure, which acts like a molecular vise, holding them in place while driving reactions forward. Lab experiments at the University of California, Irvine, confirmed this. Researchers subjected silica-rich clays to simulated desert conditions, alternating between soaking and desiccation. The result, silica not only protected amino acids from breaking down but accelerated their assembly into peptide chains, the backbone of proteins. It's as if the desert's harshness was a feature, not a bug, a natural pressurization system for chemistry. But the real magic happens in the timing. Every wet phase brings a fresh influx of molecules, while each dry phase concentrates them, eliminating the dilution problem that plagues ocean-based theories. In places like Chile's Atacama Desert, one of Earth's driest regions, scientists have found organic molecules clinging to salt and silica deposits, preserved for millennia in a state of suspended animation. When rare rains come, these molecules briefly revive, interacting in ways that would be impossible in perpetually wet environments. NASA studies of Mars-like conditions in the Atacama suggest this process isn't unique to Earth. Any rocky world with occasional liquid water and silica-rich soils could host similar chemistry. Deserts also solve another puzzle: chirality, the handedness of biological molecules. Life on Earth uses exclusively left-handed amino acids, but most abiotic reactions produce equal mixes of left and right-handed forms. Intriguingly, silica surfaces under dry-wet cycling have been shown to favor one chiral form over another, potentially explaining how life's molecular uniformity emerged. Experiments at the University of Strasbourg demonstrated that when amino acids adsorb onto silica and undergo repeated hydration and dehydration, certain configurations persist while others wash away. Over countless cycles, this could have tipped the scales toward the homochirality we see in all living things today. What makes desert theories so compelling is their brutal simplicity. No elaborate vent systems or tidal mechanics are required, just the unyielding pendulum of wet and dry, acting on materials as common as sand and sunlight. And if that's enough to coax complexity from chaos, then deserts across the cosmos, from Mars's ancient river deltas to the dune seas of exoplanets, might have been staging grounds for life's molecular debut. The universe, it seems, doesn't need gentle nurseries to build life's ingredients. Sometimes all it takes is a scorching wasteland, a fleeting rain, and the patience of stone. Comets have long been suspected as cosmic couriers, delivering water and organic molecules to early Earth. But what if they brought more than just ingredients? What if they carried the universe's first molecular blueprints? The Rosetta mission's rendezvous with Comet 67P revealed something astonishing. Its icy nucleus wasn't just a frozen lump of water and dust. It housed a sprawling network of organic compounds, from glycine to phosphorus, all trapped in a crystalline architecture that resembled a microscopic labyrinth. This wasn't random debris, it was a structured repository, with ice acting as both preserver and architect. Laboratory experiments show why this icy organization matters. When water freezes in the vacuum of space, it doesn't form dense blocks, it creates porous, honeycomb-like structures, their walls lined with trapped molecules. Under the faint warmth of distant starlight, these ices undergo a subtle alchemy. Molecules that would never interact in open space are forced into proximity, their movements choreographed by the ice's crystalline scaffolding. Researchers at the University of Hawaii simulated these conditions, exposing ice mixtures to ultraviolet radiation. Over time, the ices darkened as simple compounds like formaldehyde and ammonia linked into sugars and even nucleobases, the letters of life's genetic code. The ice didn't just protect these molecules, it guided their assembly like a molecular 3D printer. The real magic lies in how comets could have transplanted this system to Earth. As a comet nears the Sun, its surface ices sublimate, but deeper layers remain intact. A layered library of organics. Upon impact, the shock of entry wouldn't just melt the ice, it would flash heat pockets, creating temporary pools of concentrated molecular soup. Studies of simulated comet impacts reveal that these fleeting liquid zones can foster reactions far more complex than static ice, with some experiments producing short protein-like chains. Even more compelling, the Murchison meteorite, a relic of the early solar system, contains tiny pockets where water once froze and thawed, preserving organic molecules in arrangements eerily similar to those found in Comet 67P. This shifts how we view cometary deliveries. They weren't just haphazard bombardments. Each impact could have been a carefully timed infusion, releasing pre-organized molecular kits into Earth's early environment. And if comets operated as cosmic chemistry sets, then the same process might have unfolded on Mars, Europa, or anywhere with a steady rain of icy visitors. The universe, it seems, didn't leave life's origins to chance. It engineered celestial supply chains with comets as the silent couriers of complexity. Deep beneath the surface of early Earth, where scalding waters met molten rock, carbon dioxide underwent a transformation that turned it into one of nature's most versatile molecular matchmakers. Under extreme heat and pressure, CO2 doesn't behave like a gas or a liquid. It becomes something else entirely. A supercritical fluid with properties that defy conventional chemistry. Imagine a substance able to seep through solid rock like a gas, yet dissolve materials like a liquid, all while operating at temperatures that would obliterate delicate organic compounds in ordinary conditions. This paradoxical state, occurring naturally in geothermal systems, could have provided the perfect Goldilocks zone for life's precursors to assemble. What makes supercritical CO2 so remarkable is its dual nature. Unlike water, which struggles to dissolve nonpolar organic molecules or organic solvents that repel salts and metals, supercritical CO2 bridges both worlds. Laboratory studies at the University of Leeds revealed something astonishing. When exposed to mixtures of fatty acids and metal ions under geothermal conditions, supercritical CO2 acted like a molecular shuttle, stripping metals from mineral surfaces and delivering them to organic compounds, facilitating reactions that wouldn't occur in water alone. This solves a long-standing paradox in origins of life research: how key inorganic catalysts like iron and nickel, essential for modern enzymes, could have been incorporated into early organic chemistry without precipitating out as useless sludge. The real magic happens in the details. Supercritical CO2 density fluctuates dramatically with small changes in pressure, allowing it to be tuned like a universal solvent. In experiments simulating ancient hydrothermal vents, researchers watched as this tunability selectively concentrated rare molecules. For instance, nucleotide bases dissolved readily at certain pressures, while sugars precipitated out, creating natural sorting mechanisms that could have concentrated life's building blocks. Even more compelling, when fatty acids were present, the CO2 helped them self-assemble into stable vesicles, their membranes unusually resilient to the high temperatures of geothermal environments. This isn't just theoretical. Modern analogues exist in places like New Zealand's Champagne Pool, where CO2-rich fluids gush from below, creating conditions strikingly similar to those proposed for early Earth. Analyses of these fluids show they carry both organic compounds and metals in solution, supporting the idea that supercritical CO2 could have been a universal ferry for prebiotic chemistry. And if that's true here, similar processes might be unfolding in the subsurface oceans of Europa or Enceladus, where geothermal activity meets icy crusts. The universe, it seems, has a penchant for turning volatile gases into architects of complexity, given just the right combination of heat, pressure, and time. What if the story we've been telling about life's molecular origins has the sequence backwards? For decades, the prevailing view painted a tidy progression: first simple molecules, then nucleotides, then RNA, and finally proteins. But a radical theory flips this script, suggesting peptides and nucleotides didn't take turns, they built life together, in a molecular tango where neither partner led. The evidence: strange hybrid molecules called peptide nucleic acids, or PNA, which weave the features of both worlds into a single strand. Picture a molecular chimera, the backbone of a protein fused with the information-carrying power of DNA. In lab experiments, these PNA strands exhibit eerie capabilities. They bind to RNA and DNA more tightly than those molecules bind to themselves, hinting at a primordial versatility lost to modern biology. The beauty of coevolution lies in how it solves two puzzles at once. Nucleotides alone struggle to form long chains without enzymes, while peptides lack the precision for genetic storage. But together, they cover each other's weaknesses. At the University of Cambridge, researchers simulated early Earth conditions with cycles of wet and dry environments, and watched as amino acids and nucleotide precursors began linking into PNA-like structures spontaneously. These hybrids didn't just form randomly. Their peptide backbones provided stability against degradation, while their nucleobases allowed for rudimentary information transfer. It's as if chemistry had discovered a shortcut, bypassing the fragility of pure RNA or the limitations of proteins by merging their strengths. Even more compelling are the mineral catalysts that could have facilitated this partnership. Certain clays and metal sulfides have surface properties that favor the assembly of both peptides and nucleotides simultaneously. Experiments at the Georgia Institute of Technology showed that when nucleotide precursors and amino acids are exposed to volcanic mineral surfaces under geothermal conditions, they don't just coexist, they cooperate. The minerals act like molecular referees, aligning the components so peptide bonds and phosphodester linkages form in tandem. Some resulting structures even show catalytic activity, hinting at how early PNAs might have kick-started primitive metabolism without the need for specialized enzymes. The implications ripple beyond Earth. If hybrid molecules were life's true starting point, then our search for extraterrestrial life might need recalibration. Missions analyzing Martian soil or Europa's plumes could overlook PNA-like compounds if we're only looking for pure DNA or protein signatures. Meanwhile, synthetic biologists are already exploiting PNAs for medical therapies, their ancient design proving unexpectedly useful in modern gene editing. It's a humbling reminder that life's origins may not follow the linear path we imagine, but rather a messy, collaborative dance where molecules improvise their way toward complexity, one hybrid experiment at a time. Earth's moon wasn't always the distant, serene companion we see today 4 billion years ago. It loomed much closer, its gravitational pull fierce enough to warp the planet's very shape. This cosmic tug-of-war didn't just create dramatic tides, it unleashed forces powerful enough to churn Earth's early oceans like a boiling cauldron. Imagine coastal waters rising and falling not by a few feet, but by hundreds, dragging minerals from the depths and exposing shallow basins to relentless mixing. Scientists calculating these ancient tides estimate they were up to 10 times stronger than today's, transforming coastlines into colossal chemical reactors where molecules had no choice but to collide, bond, and evolve at breakneck speeds. The magic of tidal heating lies in its rhythm. Unlike random lightning strikes or volcanic eruptions, tides operated with clockwork regularity, pumping energy into coastal systems twice a day like a prehistoric metronome. Research from the University of Washington reveals how this predictability could have turbocharged prebiotic chemistry. In simulations mimicking tidal zones under extreme lunar pull, wet-dry cycles occurred faster and more frequently, compressing years of molecular experimentation into months. Amino acids subjected to these rapid cycles formed peptides up to 5 times quicker than in static pools, while RNA precursors linked into chains at rates previously thought impossible without enzymes. The moon, it seems, didn't just light the night sky. It served as a relentless stir rod for life's primordial soup, but the real game changer was heat. As tidal forces flexed Earth's crust, friction generated geothermal warmth along coastlines, not enough to sterilize but sufficient to lower energy barriers for critical reactions. Modern analogs exist in places like Canada's Bay of Fundy, where today's modest tides still cause measurable temperature spikes in tidal flats. Scale this up to ancient magnitudes, and you get environments where warmth from tidal friction could have complemented hydrothermal systems, creating thermal gradients ideal for molecular self-organization. Some models suggest these tidal hot zones might have reached temperatures perfect for facilitating the formation of lipid membranes around 50 to 70 degrees Celsius, while still protecting fragile organics from destruction. The math gets even more compelling when considering resonance effects. Early Earth rotation was faster, with days lasting just 6 hours. This syncopated rhythm, short days meeting intense tides, may have created standing waves in sheltered basins, concentrating molecules in swirling eddies. Oceanographers comparing these conditions to modern tidal resonances in Alaska's Turnagain Arm estimate that certain coves could have amplified molecular concentrations by a factor of 100 or more. Picture a natural version of laboratory centrifugation, where the ocean itself sorted and concentrated life's ingredients without any human intervention. What's often overlooked is the moon's role as a cosmic timekeeper. Its steady retreat, currently about 1.5 inches per year, means tidal forces weakened gradually, allowing coastal chemistry to adapt rather than collapse. This slow transition might have given early molecular systems the stability needed to cross the threshold from chemistry to biology. Exoplanet researchers now hunt for similar moon-planet duos, realizing that a world without a large moon might lack this gentle, persistent stirring mechanism. The more we study our own lunar partner, the more it seems its greatest gift wasn't just the tides but the patience to let them work their magic over billions of years. Among the most dramatic features of the deep ocean floor, black smoke vents stand like towering skyscrapers, spewing superheated plumes thick with dissolved metals. These mineral-laden chimneys, reaching temperatures over 350 degrees Celsius, might seem like the last place life's delicate molecules could emerge, yet their very violence could have been key to organizing chemistry. Unlike their gentler alkaline cousins, black smokers are chaos incarnate, ejecting clouds of iron, copper, and zinc sulfides that crystallize into porous structures as they hit frigid seawater. It's in this turbulent interface where scalding meets freezing that nature may have forged its first enzyme-like catalysts. The secret lies in the smoker's metallic bounty. Modern enzymes rely on metal clusters at their core to shuttle electrons and stitch molecules together, and black smokers offer these same elements in raw, abundant form. Lab experiments simulating vent conditions show something extraordinary. When hot, metal-rich fluids mix with colder water, they spontaneously form microscopic chimneys with intricate internal pores. These cavities, lined with iron-nickel sulfides, bear an uncanny resemblance to the active sites of contemporary enzymes like ferredoxin and hydrogenase. Researchers at the Carnegie Institution watched as these mineral structures facilitated reactions identical to those performed by living cells, reducing carbon dioxide, splitting hydrogen molecules, even stitching together short amino acid chains. It's as if geology had stumbled upon biochemistry's blueprints billions of years before cells existed. But the real breakthrough came when scientists examined how these mineral catalysts self-organize. Under electron microscopes, vent-derived metal sulfides show layered structures with precise spacing between atoms, almost like molecular scaffolding. When organic compounds like pyruvate or acetate encounter these surfaces, they don't just stick randomly, they align along the mineral's crystalline grooves, positioning themselves for specific reactions. Some studies even suggest these mineral templates could have guided the formation of peptide sequences, acting as primitive versions of the ribosomes that build proteins today. The Mothra vent field near the Pacific's Juan de Fuca Ridge offers living proof. Its chimneys host microbial communities that still exploit these mineral catalysts, their biochemistry echoing processes that might have birthed life itself. What makes black smokers uniquely potent is their dynamic instability. Unlike static clay surfaces or ice grains, these vents are constantly rebuilding themselves, collapsing and reforming in cycles that mimic metabolic turnover. A single vent might spew metal particles that catalyze a reaction, only for those particles to dissolve and re-precipitate downstream, refreshing the catalytic surface. This natural recycling solves a key problem in prebiotic chemistry: how to maintain active sites without them becoming clogged or degraded. Hydrothermal systems at the Lost City field demonstrate this elegantly. Their carbonate towers, though chemically distinct from black smokers, show similar patterns of dissolution and regrowth, hinting at a universal principle of self-renewing catalysis. The implications extend beyond Earth. Jupiter's moon Europa likely harbors similar metal-rich vents beneath its icy shell, as suggested by the iron compounds detected on its surface. Meanwhile, Mars' ancient seafloors show fossilized hydrothermal deposits rich in the same sulfur minerals that power black smoker chemistry. Whether in our solar system or beyond, these violent, mineral-charged systems may represent nature's favorite crucibles, places where raw elemental energy and precise atomic geometry conspire to turn chaos into complexity, one catalytic puff of superheated metal at a time. Hydrogen cyanide, a molecule synonymous with toxicity today, might have been the unsung architect of life's genetic code. In Earth's primordial skies, this simple compound, just 3 atoms linked like a molecular tripod, could have accumulated in concentrations that would stagger modern chemists. Unlike today's oxygen-rich atmosphere that breaks HCN apart, early Earth's air, a thick cocktail of nitrogen, carbon dioxide, and volcanic gases would have acted as a preservation vault, allowing hydrogen cyanide to persist and react over millennia. The key lies in its structure. That carbon-nitrogen triple bond is a coiled spring of chemical potential, ready to snap open and latch onto other molecules with startling efficiency. Laboratory experiments reveal just how versatile this unassuming molecule can be. When researchers at the Georgia Institute of Technology simulated early atmospheric conditions with HCN and subjected it to UV light or electrical discharges, something remarkable happened. The cyanide molecules began linking into chains, folding back on themselves to form rings, and not just any rings, but adenine, one of the 4 nucleobases found in DNA. This wasn't a rare fluke. Under the right conditions, the reaction pathways from HCN to adenine are almost mathematically inevitable, like a molecular origami that folds itself given enough time and the right triggers. Even more astonishing, the same process can produce guanine, another critical nucleobase, through slightly modified versions of these reactions. But how did such a toxic substance accumulate in meaningful quantities? Here's where early Earth's geology becomes crucial. Volcanic eruptions would have belched vast amounts of hydrogen cyanide into the air, while impacts from carbon-rich meteorites could have delivered it preformed. Unlike today, where HCN quickly disperses or reacts, the planet's reducing atmosphere devoid of free oxygen would have allowed it to linger, pooling in low-lying areas or dissolving into shallow seas. Computer models suggest that in certain regions, particularly near volcanic basins or impact craters, HCN concentrations might have reached several % of the local atmosphere, a far cry from the parts per billion levels we associate with cyanide today. The real genius of this system lies in its scalability. Hydrogen cyanide doesn't just form nucleobases, it's a molecular multitool. Under slightly different conditions, say, in the presence of formaldehyde or ammonia, HCN can assemble into amino acids like glycine or even the ribose sugars needed for RNA. NASA Ames Research Center demonstrated this by irradiating icy mixtures containing HCN, watching as they transformed into a veritable buffet of prebiotic compounds. It's as if the universe encoded a backup plan into this molecule, a single starting material capable of branching into nearly every major component of life's machinery. What makes this theory so compelling are the cosmic breadcrumbs we've since discovered. The Murchison meteorite, a 4.5 billion-year-old relic from the solar system's infancy, contains not just adenine and guanine, but their HCN-derived precursors. Meanwhile, telescopes have spotted hydrogen cyanide swirling in the atmospheres of Titan and in the molecular clouds where new stars form. The implication is profound. This toxic molecule might be a universal feedstock, quietly assembling life's blueprints wherever planets coalesce. Yet the story doesn't end there. Recent work at Harvard has uncovered an even more elegant twist. HCN's reactions might have been guided by mineral surfaces. When hydrogen cyanide encounters certain clays or metal sulfides, its transformation into nucleobases accelerates dramatically, with some minerals favoring specific products. Imagine volcanic coastlines where cyanide-rich waves lapped at mineral-laden rocks, each interaction editing the molecular chaos into something increasingly refined. It's a reminder that life's origins weren't a single eureka, a moment, but a symphony of chemistry, geology, and time, all orchestrated by one of the universe's most unlikely maestros. At the coldest edges of the universe, where temperatures hover just a few degrees above absolute zero, molecules perform what seems like magic. They pull off chemical reactions that should be impossible reactions that, by all known rules of chemistry, require heat and energy to occur. The secret lies in one of quantum mechanics' most bizarre phenomena, tunneling. Picture a molecule trapped behind an energy barrier, like a ball sitting at the base of a mountain. In classical physics, that ball needs to climb over the mountain to reach the other side, an impossible feat in the frozen depths of space. But in the quantum realm, particles don't always go over barriers, they slip through them, vanishing on one side and reappearing on the other as if the mountain never existed. This isn't just theoretical. Observations of interstellar clouds reveal molecules like hydroxyl radicals and formaldehyde forming at rates that defy conventional chemistry. Laboratory experiments at the University of Leeds demonstrated how tunneling allows hydrogen atoms, the lightest and most quantum mechanical of elements, to hop between molecules even at 10 degrees Kelvin, a temperature where all classical reactions grind to a halt. In one striking experiment, researchers watched as hydrogen swapped places between methane and an organic radical, nearly 100 times faster than classical models predicted. The atoms weren't colliding with enough energy to react, they were teleporting through the energy barrier itself. What makes quantum tunneling so revolutionary for origins of life research is its implications for prebiotic chemistry. Many of life's key reactions, like the formation of water from hydrogen and oxygen, or the assembly of simple sugars, require just the right amount of energy. In the cold vastness of space or Earth's early oceans, that energy was often in short supply, but tunneling could have provided a backdoor, enabling reactions that otherwise would have taken billions of years to occur. Some researchers now speculate that RNA nucleobases, notoriously difficult to form without heat, might have stitched themselves together through quantum shortcuts in icy cometary grains. The most tantalizing evidence comes from spectral readings of molecular clouds like Sagittarius B2, a star-forming region 25,000 light-years away. Astronomers have detected complex organic molecules there, ethanol, vinyl alcohol, even glycolaldehyde, in abundances that classical chemistry can't explain. Quantum tunneling provides the missing link. Simulations at the Max Planck Institute show that with tunneling factored in, these molecules form orders of magnitude faster, matching astronomical observations almost perfectly. This reshapes our understanding of life's ingredients. If quantum effects allowed critical reactions to proceed in the freezing void of space, then the building blocks of life might have been assembling long before Earth formed, hidden in the icy hearts of comets and asteroids. It's a humbling realization. The same spooky action at a distance that baffles physicists today could have been the silent architect of complexity all along, an invisible hand guiding molecules toward life in the coldest dark. From Earth's churning hydrothermal vents to the frozen depths of interstellar space, the story of life's first complex molecules isn't a single thread. It's a tapestry woven from 30 astonishing possibilities. Some theories root life's origins firmly on our planet, where tidal pools concentrated ingredients, volcanic glass sculpted them, and radioactive beaches provided energy. Others cast the net wider, suggesting comets delivered pre-assembled molecules or that quantum tunneling in icy space clouds bypassed chemistry's usual rules. The beauty lies not in choosing one story over another, but in realizing how many paths the universe might have taken and perhaps did take simultaneously. Take hydrothermal vents and cosmic dust grains, seemingly opposite environments, yet experiments show both can produce amino acids under wildly different conditions. Iron-sulfur minerals in vents catalyze reactions through brute force geothermal energy, while delicate ice coatings on interstellar grains rely on patient UV radiation. The truth might be that life's building blocks weren't forged in a single cosmic kitchen, but in countless ones across the galaxy, each contributing unique flavors to the primordial recipe. Even on Earth, tidal pools could have assembled RNA strands while nearby radioactive sands stabilized them, all while atmospheric hazes rained organic material from above, a convergence of processes that together tipped the scales toward biology. This multiplicity of pathways reshapes how we search for life beyond Earth. If one environment fails on an alien world, say, no hydrothermal vents exist, others might still succeed. Mars' ancient deserts could have hosted silica-driven peptide formation, even as its vanished oceans brewed alkaline vent chemistry. Europa's subglacial seas might combine tidal flexion with comet-derived organics, creating a molecular symphony unlike anything we've seen. The universe, it seems, hedges its bets, exploring every conceivable route to complexity. So which theory resonates with you? The elegance of RNA self-assembling in evaporating pools, the drama of meteorites delivering phosphorus like cosmic UPS trucks, or the quantum weirdness of molecules tunneling through energy barriers in deep space? Drop your favorite in the comments. Let's debate which pathway feels most profound, or better yet, how they might intertwine. And if you're hungry for more cosmic mysteries, hit subscribe to journey with us into the next frontier of scientific discovery. After all, the universe's greatest stories are still being written one molecule, one theory, one aha moment at a time.